Welcome to our channel Biology Classes. Today we will discuss about plasma membrane. As we all know that plasma membrane is also known as cell membrane. It is a biological membrane which separates and protects inferior of cell from outside environment. It is a dynamic and fluid structure. It acts as selectively permeable membrane. This membrane will decide uh, which foreign substance will enter in the cell or not. Okay, it passes only selected substances. This structure was explained by two scientists, Singer and Nicholson, in 1972. They gave name of fluid mosaic model to this plasma membrane structure. If we talk about chemical constituents of plasma membrane, ratio of protein to lipid varies depending on cell type. According to cell type, ratio of protein or lipid varies. If we can see example of human RBC. In human RBC, plasma membrane contains 43% lipid and 49% protein, whereas plasma membrane of mouse liver cell will contain 54% lipid or 46% protein. As we can see this structure of plasma membrane, this is lipid bilayer structure, and this is hydrophobic. Hydrophobic means water phobic. uh, Upper part of this lipid is polar region, and this one is non-polar region, this tail. And this structure is this is integral protein. Some proteins are fully integrated in these bilayers. They are called integral proteins, whereas some proteins are present at peripheral region. Those proteins are called peripheral proteins. If we talk about glycoproteins, this is glycoprotein structure, which is attached with this integral protein. Glycoprotein, what are glycoproteins? Means carbohydrate. Glyco means carbohydrate. Carbohydrate attached with proteins called glycoproteins. As we talk about carbohydrate present in plasma membrane structure as in two forms, glycoprotein or glycolipids. Glycoprotein means we know glyco means carbohydrate attached with protein. This is glycoprotein. And if carbohydrate attached with lipid, so they called glycolipids. Carbohydrate present either as glycoprotein or glycolipid in this structure of plasma membrane. One more thing, why these scientists named it fluid mosaic model. They called it fluid model because these lipids and proteins are in motion in this plasma membrane structure. And why they called it mosaic, because these lipids are arranged in a manner, uh, so they called it mosaic model. Now move forward to our next topic, phospholipid bilayer. The basic structure of plasma membrane is phospholipid bilayer. It is composed of two leaflets of amphipathetic molecule, phospholipid molecules. Uh, what does it mean? It means amphipathetic means both polar and non-polar region. Here is a diagram of phospholipid. This is head part and this is tail part. Uh, head is a polar region and tail is a non-polar region. Types of phospholipid. If we break this term into two parts, phospho plus lipid. This means phospho represents phosphate group and lipid we know fatty acid chains. Okay? Types of phospholipids are glycerophospholipids, sphingophospholipid. What do you understand by glycerophospholipid or sphingophospholipid word? To understand this word, if we break down into three terms, glycerophospholipid. Refers to glycerol, phospho for phosphate group, and lipid for fatty acid chain. Means glycerol is a backbone of this structure, and two fatty acids attached to this glycerol, and phosphate group is also attached to this glycerol, and alcohol or either nitrogenous bases will attach to this phosphate group. This is whole structure of glycerophospholipid. Sphingophospholipid. Sphingo means sphingosine, phospho means phosphate group, and lipid for fatty acid. There is a one backbone sphingosine, and single fatty acid is attached to this backbone, or phosphate group is also attached to this sphingosine, and alcohol is attached to phosphate group. Alcohol are choline, serine, or ethanol, amine, or myelin. These are alcohols. Choline is present as an alcohol, the structure called phosphatidyl choline. If serine is present as alcohol, the structure called phosphatidyl serine. If ethanol amine present, then structure will be called phosphatidyl ethanol amine. If myelin is present, sphingomyelin the structure. There is a net charge present on these phospholipid structures. Let's say there are net charges present on phospholipids, positive charge, negative charge, or neutral charge. Positive charge present on phosphatidyl serine, phosphatidyl glycerol, cardiolipin. Neutral charge present on phosphatidyl choline. There are some rare phospholipids that contain positive charges. As we already talked about it previously, glycolipids are found in outer leaflet of plasma membrane with their carbohydrate portions exposed on the cell surface. Glycolipid, glyco means sugar residues, and lipid, sugar residues attached to lipid, the term glycolipid. It is derived from either glycerolipids or sphingolipids. Sugar residue may be glucose or galactose. There are two types of glycolipids, simplest form of glycolipids or complex form of glycolipids. Simplest glycolipid, which is generally known as cerebroside, which contains single sugar residue, and complex type of glycolipid is gangliosides, which contain 7 residues of sugar, and there is a branching present in the structure of ganglioside, which make it more complex. Now sterol. What is sterol? Sterol is a structural lipid which is also present in plasma membrane. Basic structure of sterol is a steroid nucleus consisting of four fused rings, 3 with 6 carbon and 1 with 5. If we see this diagram of sterol, 1, 2, 3, 4, 5, 6. 6 carbon ring, 1, 2, 3. 3 rings. 3 rings are of 6 carbon, which is fused. Okay, one ring, 1, 2, 3, 4, 5. One ring is of 5 carbon. This is the structure of sterol. Cholesterol, which is major sterol present in animal cells, but in plant cells there is a lack of cholesterol, so another sterol is present which is called stigmasterol, and whereas in bacteria there is no sterol present, so they lack sterol. Now motion of lipid in plasma membrane will be continued in next video. Thank you. Like, share and subscribe our channel.